Alright, so I want to talk a little bit about this book called The Art of the Soluble. This is by Peter Medawar, who won the Nobel Prize in 1960 for his work in medicine and physiology on artificial organs and things like that. And this book was written around the time he won the Nobel Prize, perhaps a little bit before. It's an interesting book because it's sort of it reprints a bunch of different books and gives his insights on those books. So, he was very strangely anti-Darwinian, or you get the sense that he was trying to criticize Darwin for the sake of criticizing Darwin. He wasn't a creationist, but he was like kind of, maybe the better way to put it is a contrarian. So that's kind of where, when I go through the book, this is kind of the frame that I want you to keep in mind. Solubility is this phenomenon in chemistry where if you add some substance to, say, something like water, it will sit in solution at certain concentrations, but at a critical concentration, it will become saturated and it will fall out of solution. And so in this case, you have sugar in water. If you add some sugar, you get sugar that's dissolved in water. It's suspended in the water. And then eventually, if you add more, enough sugar, you can get precipitate out on the bottom in this sugar solution that's sitting on the bottom, and then the deposited sugar becomes greater and greater. So that's a reference to the title. And then this is an example of D'Arcy Thompson's work. So he focuses on D'Arcy Thompson's On Growth and Form book. And this is a book that was published in 1917. So we're going to do a very historical view here. We're going to view a book that was published in 1958, talking about a book that was published in 1917. And so keep that in mind when we talk about these things, because of course we're sitting in year 2025, and it's very different from these other time periods. So we're kind of like looking through the lens of two other time periods in the past where they didn't have certain concepts that we have today. They had, different ways of thinking about biology and mathematics and things like that. So just keep that in mind. And then this is D'Arcy Thompson's main idea in On Growth and Form, which is that you can take organisms and you can plot them out on these grids, and you can describe the morphology by looking at the grid and then looking at related forms and shrinking this coordinate system or warping this coordinate system accordingly. So you see that we have these different shaped, I guess they're crustaceans or they're some sort of insect. They're carapace, and their carapace has different forms and shapes, and so they're being transformed. The initial example here is just a square Cartesian coordinate system, a grid, and then that grid is warped and deformed as the different landmarks change their position relative to the original organism. So you see on the left, there's this original reference organism, and then to the right, you have these other forms that progressively change, and you get these differences in the grid. And the grid, those transformations in the grid then allow you to mathematically describe or quantitatively describe those changes. So looking at sort of the antenna, some of these organisms have large antennae, some of them don't have any, and so that's going to affect the size of the grid. So this is the idea that D'Arcy Thompson brought to the fore. It was very early theoretical biology, and at the time, of course, they didn't have computers to work these problems out, so a lot of it was hand-drawn. And it's an interesting book on growth and form. My take on it is that it's mathematical biology. A lot of people think it's more mathematics than biology. But what's interesting to remember is that it's basically a description of form or shape without necessarily thinking about the evolutionary biology or even the developmental biology to some extent, because he does focus on, this sort of mathematical interpretation of life. And so we'll get into this in a minute. So it's also important to remember, like I said, we're dealing with history, we're dealing with different points in time. Now, what we're talking about with Medawar's book and then D'Arcy Thompson's work is that there was no vocabulary in physics for complex phenomena until the latter half of the 20th century. So we're familiar with terms like emergence or nonlinearity. Those weren't really commonplace in physics even until later in the 20th century. The 60s and 70s, we started to get these terms emerge, and they describe things that they also talk about here, but they don't have the words for them. So I think that's a very important point. When you have some phenomena you're looking at, it's much easier to talk about it when you have the vocabulary than when you don't. If everyone has to kind of describe it in their own way and hunt around for the words and hunt around for the sort of mental model that they need to understand what's going on, then it's much harder to really give like a unified theory or a unified account of what's going on. And this is a problem in biology as well. So people often in biology, complain that something's too mathematical or something like that. It's not real biology. But it's important to remember as well is that there's really no vocabulary that's biology specific for complex phenomena, even today. A lot of the vocabulary that we use to describe things like emergence and nonlinearity in biology are borrowed from physics. And so biology being its own domain maybe requires its own language or vocabulary of complexity as opposed to just simply borrowing from physics. Those are all important points that we're going to get to in a moment. So this is Art of the Soluble, published first in 1958. And so that's the era we're talking about. This book, it's not that thick, but it's a reproduction of several books with Medawar's critique and his comments embedded. This book serves as a critique of biological theory up to that point. So he talks about Spencer, he talks about D'Arcy Thompson, he talks about Darwin a lot. He was kind of bizarrely anti-Darwinian. If you read it, it's not that he was a creationist, but you have to remember that Darwin, when he published on Origin of the Species, there were a lot of people who, were not really accepting of it right away. And, you had later developments which kind of culminated into, say, like the neosynthesis, which didn't come until like the 1930s or 1940s. And it was well known that the German embryologists, for example, were not Darwinians. In fact, they didn't accept Darwin's Origin of the Species at all. They had their own sort of agenda, and they went with that. And, D'Arcy Thompson, being a morphologist, being interested in development, also didn't really use Darwin as the basis for a lot of his things. And so it can be interpreted sort of as maybe non-biological, when in fact it was sort of pre-Darwinian work, in truth. But Medawar also had, was kind of, I don't know, he was anti-Darwin. He was very critical of Darwin, both in this book, Art of the Soluble, kind of directly addressing, things about, I think there's a chapter called Darwin's Illness, which is about like Darwin's preoccupation with sex or something like that. To, concepts that, didn't necessarily square away with what Medawar was thinking. So in any case, he does focus on this book on growth and form. This was first published in 1917. It was a sort of a comment, but it was also 1,000 pages long. So there's no shortage of comment there. And so D'Arcy Thompson, of course, was interested in describing development, describing morphology, and describing changes in morphology. And of course, we can do that with, evo-devo today. We can do that with evolutionary theory. We can do that with computational biology and development. But he had none of those things. And aside from that, we didn't have computers, so people really had to visualize things on pad and paper, and they had to draw things out. And so this is kind of an attempt at putting mathematics or mathematical cast on this. So, that's kind of where he's coming from. He actually had two conceptions of biological science. So he kept sort of the evolutionary, which they call Darwinian frame, which was the ultimate level of causation, separate and apart from the anatomical or proximate frame of causation. So if you look at the anatomy of an organism, it gets there through development. That's different from evolution, which is the ultimate causation. And I know that, if you think about evo-devo, that's actually not the way they think about biology, that those things are connected. But this is the way they thought about this. And it's interesting that Nico Tinbergen, who was not necessarily a contemporary, but also published on this in the context of behavior, talked about two levels: development, which is proximate causation, and evolution, which is ultimate causation. And this has to do with behavior. How do you acquire behaviors? Is there's this nature-nurture distinction, but there also is this distinction between, like behaviors that you acquire within the lifetime and behaviors that you see across generations. So it's interesting. But Thompson took a molecular and an anatomical view of biology. He focused on the anatomy. He doesn't mention like gene pathways or any of that other stuff that we talk about in evo-devo. But he did have this sort of molecular view of life, such as it was back in 1917, and an anatomical view. So it's interesting that that's mentioned in the book. And so if we take some of these ideas and put them on a grid, we have proximate causes and ultimate causes. We have molecular causes and anatomical causes, or rather, things that are expressed in molecular terms and things that are expressed in anatomical terms. So this is kind of the framework that we can think about D'Arcy Thompson's work. Now, D'Arcy Thompson gives an example in his book about how, some biological structures are maybe not the product of evolution. And so he talked about an example of the trabecular bone. So trabecular bone is a specific type of bone in the body. He talked about it as kind of being this thing that grows in response to function. So in the case of trabecular bone, is it an adaptation, a product of evolution that gets put down, in organisms that have acquired trabecular bone as a trait? And then, it just kind of like is shaped by fitness and things like that. Or is it an adaptive material, which takes on its shape in response to function? So in other words, if, I'm an organism that has very fast movements, or if I'm an organism that needs a lot of reinforcement in my body, I grow my trabecular bone in ways that is different from an organism that doesn't have those things. And, you might say, well, that's obviously, a product of, it's an adaptation to the environment. It's something that we can just account for through evolution. But, you can also look at examples where there's a lot of variation within what we call life history. And so this is kind of, showing D'Arcy Thompson's distinction between what's going on in development or in life history and then what's going on in evolution, and using evolutionary adaptation as a descriptor of that. So it's about assigning sort of the cause, but weirdly there's no experiment here to demonstrate this. It's just kind of like I'm observing morphology and then I'm making decisions based on that, and making kind of mental models based on that. So in his example, the remodeling and regeneration of trabecular bone enable an environmental response, but of course, so this is only up to a point, and this is something whereas today we call facultative adaptation, which is you have an adaptation that's a response to your interactions with the environment, and you can develop these facultative adaptations, and they look very much like evolutionary adaptations, but they're not. Another possibility, of course, that D'Arcy Thompson didn't talk about is this idea of evolutionary encoding. So this is, what would later be sort of the standard explanation in evo-devo, where you have this evolutionary encoding. In other words, you have these, these life history pressures, these interactions with the environment, and they get encoded in terms of heredity. So they get encoded in terms of being encoded in genes. So this is, of course, where maybe like forces shape the limbs of ancestors. This translated into some sort of developmental instruction that was inherited in a Mendelian form. So, again, you have a bunch of variation in the ancestors. Those things get selected for, or sometimes, you could have traits for plasticity that get selected for. And you basically end up with these developmental instructions that result from the fittest forms being selected by natural selection. And then, that's what you have. So what D'Arcy Thompson was observing was the end product of this evolutionary encoding. It wasn't that facultative adaptations are predominant and that evolution is irrelevant. So, again, when you have the right vocabulary, when you have the right mental model for this, it makes all the difference in the world in terms of the explanation that you use. Another aspect, and maybe it's very telling about D'Arcy Thompson's work, is that he had a pre-scientific view of development. So that pre-scientific view was often two schools of thought. One was this idea of preformation, which is, that should read preformation, which is where you have ready-made instructions. So it's like you have this little embryo that's going to form into a human, and it's just a matter of growing, and it's the instructions are already in there. You just need to bake the embryo. And of course, we know that that's not really the case. That's not that's not really the way things work. And that was contrasted with epigenesis, or fulfillment, where there was a sort of generative process by which the embryo achieved its form. So, but this is still all pre-scientific. This is not like, the way we understand things today. So we use the term epigenesis, but it's quite different than the context in which they used it then. And then, something that Medawar says is that D'Arcy Thompson mistook the context of physical and mechanical forces. And so this idea that I pointed out here, where we have this environmental response versus an evolutionary encoding, he didn't really understand the context of what he was looking at. When you look at something, things get there in a certain way, and you have to understand that context before you can make, kind of a theoretical judgment. And so physical and mechanical forces, are they acting in life history, and then that's the thing that determines the phenotype? Or is it that the phenotype is pre-adapted, so to speak, to these physical and mechanical forces? So like deep sea fishes, for example, have these very exotic morphologies. Is it that they've shaped these exotic morphologies in development persistently? Or is it that they've evolved to these exotic forms to deal with the extreme pressures and temperatures and lack of light of the deep sea? And so this is the context of organisms or more specifically physical and mechanical forces on organisms, that Thompson, maybe didn't understand. And so this is interesting because there's been later work in this area by people like Adrian Behan and Norman MacLeod and others who have, kind of worked on this problem of morphology and physical forces and things like that. And they've come up with some interesting ideas, but again, are they understanding the context properly? People, who have backgrounds, say, in mechanical engineering or in mathematics, would they understand the proper biological context? Another part of this, and this is part of D'Arcy Thompson's book, there was a chapter called A Principle of Heredity. So this is something where Darwin and Mendel had a principle of heredity. Actually, Darwin borrowed Mendel's principle of heredity, but Darwin said we needed a principle of heredity. Thompson did not have this, and this was kind of key, is that there was no principle of heredity. He didn't really accept Darwin's, and he didn't really talk about Mendel at all, but he did talk about sort of how, development and evolution are separate. And so that makes sense that, like, if you decouple development and evolution, you're not thinking about heredity as sort of the bridge between them. And so, another thing he kind of thought about was he considered physical agencies as the basis for natural selection. So that might be like where you have a set of forces in the environment and those govern the fitness. Are you fit enough to be physically viable in a given environment? This still isn't a principle of heredity, but it basically is the way you view natural selection, such as it was. In this case, natural selection being the things that drive developmental plasticity. So Thompson had a philosophy of living organisms. So basically, physical laws correspond to organisms. The organisms that we observe are the products of physical laws. We know that, like, genes code for things. He didn't talk about genes in the book, but he did talk about these physical laws being the constraints on the shapes that organisms had. And then, of course, he used these grids to describe those changes. And you could have, of course, these deformations that could be described with variations on this grid. And, of course, you could map that to physical laws. You could map that to the effects of gravity or forces or stresses or strains or things like that. And then the second part of the philosophy of living organisms was that living organisms do nothing to contravene laws. So they cannot overcome the laws of physics. And so organisms are totally constrained by physics and therefore can be described as physical sort of entities. In which, we know that, like, organisms have to obey physical laws, but I think he was thinking in a more deterministic sense than that. Now, ironically, he wasn't a physicist. He was just using a lot of mathematics to describe life, but that's kind of that is his philosophy of living organisms. Now, he also had some thoughts on things like rate of growth, which is, of course, now today known as allometry. And so, and Medawar points out that the way Thompson thought about the rate of growth is very different than the way that, say, Huxley thought about differential growth, or that we use log scaling in modern allometry to describe sort of the difference in rate of growth between two parts of the organism. So in allometry, we might look at head size versus body size, brain size versus body size, snout length versus tail length, or snout length versus body size. Basically, the idea is that there are certain parts of the body that grow faster and at a greater rate than other parts of the body, and that's a product of evolution. And we can see that across species, and that can describe evolutionary processes, evolutionary change across different species in a clade or in a comparative context. So, you have to think in terms of Thompson's approach to rate of growth. He thought about this as literal growth, where as opposed to sort of Huxley and then the allometric school, where they think about logarithmic growth. And so you have to normalize growth. And that's how we approach scaling today. But Thompson actually approached this from a literal context. And you can tell because he's using grids that he's not using any sort of log transformation. Instead, he's using sort of talking about the literal growth of the organism on these grids that then sort of normalize the rate of growth across organisms. So the first thing here at this point is log scaling leads us to this description of differential growth that started with Huxley's work and then kind of moved on to allometric scaling. But another way Thompson thought about it that was strange, at least to the modern eye, is that he thought about growth as being this sort of compound interest. And so, it's kind of like almost like an economic metaphor, where you invest in some growth and then you get a return on that investment and you invest in more growth and so on and so forth. And the way he would describe it, say in a bivariate context, was to have sort of a first derivative versus a second derivative. So the first derivative would be rate of growth. The second derivative would be acceleration of growth, kind of like the equations of motion. And then you could describe growth in that way. Another thing he was very interested in was this concept of homeomorphism, which is something that, the cyberneticists would later use to describe the mappings between models and the environment, or models in a system. So the idea is that you have this real world, in this case, a real world of forces acting upon an organism, and you have to map that to some representation, which is the organism's shape. And so the shape of the organism describes this homeomorphism. And if you compare two shapes, you can see this homeomorphic map where there's an organism that is basically the same underneath. It's just described by two sets of forces acting upon them. So the forces provide this homeomorphic map where everything is roughly equivalent between the two organisms, and the only thing that's different are how forces act upon them, and so you can describe that difference. So thinking a little bit about Thompson's transformations on these grids, these are Cartesian grids. So, we're used to thinking in terms of fancy geometries and dimensionality reduction when we think about modern machine learning and modern sort of mathematical biology. But in this case, he just used Cartesian coordinates. And maybe that was a brilliant stroke of genius on his part to simplify things. Maybe it was woefully inadequate. But I think, I don't think it matters too much because the principle is the same. He's just describing these morphologies using a coordinate system, and he's interested in the transformations, and that's the key. But the other thing that was unique about his way of thinking about this is that he thought of the whole organism. He thought about the whole organism as everything as being important. So it was a holistic approach instead of a reductionist one. And you have, of course, different traits as landmarks on the grid, but really all of those landmarks change sort of relative to one another. So it's kind of an interesting point that you're not isolating different traits and you're taking them one at a time. He's just analyzing the whole thing all in one shot. And so if we go back to this point I was talking about with having a vocabulary for complexity, this is kind of thinking about how to measure emergence or emergent phenomena. So all of these traits change together due to physical forces. Those physical forces then force us to think about the organism not as a collection of traits, but as this emergent phenomena that results from those constraints acting upon the organism. Now, of course, this doesn't take into account things that are shaped by genetics or shaped by other things that aren't physical forces. But this is a good, I think, example of where he's basically looking at emergent phenomena without mentioning it, because he didn't have the vocabulary. So, the way he's thinking about this is that you have an embryo that is roughly similar in terms of shape from organism to organism, and then through development they acquire these different organismal shapes that are related but different. And so he thought about, common ancestry. That was part of his mental model. He thought about how embryos are basically the same, which is not really true. We know that now. And then, of course, we had these changes across organisms from sort of a common ancestry. And, again, not worried about the genetics, not worried about other changes. The only thing he's worried about is the morphology. This is an example of two different morphologies on a grid that's been deformed. So you have a square grid with this pelvis of Stegosaurus, and then you have the pelvis of Camptosaurus, which changes it. There's some changes in the way that it's, the shape, and then that's represented by changes in this grid in how the different points are aligned. And so you can describe that mathematically pretty easily. And then that describes the difference between Stegosaurus and Camptosaurus. So the last thing I want to talk about is actually going farther afield from this and talking about Valentino Breitenberg's work on this book on the texture of brains, which came out in 1977. So Valentino Breitenberg, in the Saturday Morning Neurosim group, we've been talking about his work in neuroanatomy, his work on, thinking about anatomical structures, and then linking that to sort of these artificial brains or artificial simulations. And so he had this chapter on the texture of brains called Physics and Antiphysics. And so physics and antiphysics basically is about setting this vocabulary agenda for neuroscience in particular, but also for sort of a biology-specific vocabulary for talking about things like emergent phenomena and other types of capturing phenomena in, say, like things like connectomes. So a couple words here. He views causal explanations as sort of the difference between sort of this reductionist view where we look at the level of particles. They could be molecules, they could be atoms, so you take a very atomistic view of causality. You're looking at the interactions between particles or the causal relationships between particles. As opposed to things where you have emergent phenomena where you have to describe things as in their whole to get a proper representation of their causal context. And he calls these explanations out of context. So the context of these particular causal mechanisms will give you one answer. And then looking at the whole system, which is the product of emergent phenomena, looking at causality at that level will give you another answer. And so this is very much in line with the way we view emergence today, that emergence is fundamentally different from the atomistic reductionist view. Some words about information. So he's interested in the brain, and he's interested in how you have these units of information or these units of interaction in the brain. And you can use all sorts of different units of explanation for how things happen in the brain. As it turns out, the brain consists of a few types of cells, and they usually are found together, and we can use these motifs to get there. So we have these clusters of neurons where you have like a neuron or a pair of neurons, some glia and some astrocytes surrounding them, and that's one of these units of information. And if you look at that neuroanatomically, again at the anatomical level, you can treat those units as motifs, much like we treat words or codons as motifs, or maybe the common parlance as tokens in a model. So in the analysis of DNA, we'll use codons to understand what DNA does. It turns, it gets transformed into amino acids and then into proteins, or you might have words, or sometimes people will use the term engrams to describe, things that will lead to a vocabulary, things that will lead to sentences and texts and things like that. So this is all kind of, a means of taking the whole, breaking it down, and then bridging that gap between sort of the emergent phenomena that we observe as the whole sort of organism or the whole brain, and then these particular atomistic descriptions. And so then, these neuronal cell processes consisting of a couple of neurons, some glia, some astrocytes, these are geometric relations or networks between neuronal cell processes. So what I think Breitenberg is describing here is sort of like this symplectic approach to networks that we have today, where you have different motifs in the network. You have these, first of all, you have these motifs as the nodes, which are, these sets of neurons, glia, and astrocytes, which form a fundamental unit of function that allows you to process information. But then you have these connections between them, and those connections form sort of geometric relations. They're both geometric in terms of their anatomical relative anatomical location, and they're geometrical, so in terms of the network where you can draw these shapes in the network, you can draw polygons, you can draw other types of shapes, and they are all sort of descriptive of the topology of the network. So it describes sort of how these neuronal cell processes are related, not just in terms of their how many connections they have, but also in terms of their physical co-location or their orientation to one another. And so this is where causal explanations of different types converge. So this is where we can get a sense of these two different levels of explanation and bring them together conceptually. Now, it doesn't necessarily help that Breitenberg didn't have the language for describing, we don't really have a good language for biological emergence, but we could probably do that and be better off here. We could get a better account. So, what I've presented here are all these ideas, these precursors to modern complexity theory and biological complexity. And so I hope this gives people food for thought in terms of how to approach this stuff, and maybe some of our historical antecedents that maybe will, give them a better sense of how to think about these problems. All right, so this is, I worked up this set of images based on our discussion about the problem of relative growth versus the problem of absolute growth. So the problem of relative growth goes back to Huxley, and we use this often in allometry studies. And this is where we measure one trait versus another trait. So we might measure head length with respect to body length. If we're looking at a fish, which we'll see in a minute, can be represented in other ways. So we have this proportion of the head length to the body length, and you can see this in this. You can see this in this bivariate graph here, where we take the head length and the body length of many different species or many different specimens, and we plot them out on this plot, and we get some sort of function that describes it. And a lot of times this function is logarithmic. I put a linear function here, but a lot of times it's logarithmic. So, you have at the beginning you have the linear phase, and then you have the logarithmic phase coming off at the end, where the growth essentially of the head saturates relative to the body. So this is relative growth, and the idea behind this maps very neatly onto the notion of something like Hox genes, or something like where you have different proportions of different body sizes that are selected for an evolution. So this is relative growth. But what D'Arcy Thompson was talking about was absolute growth, or the problem of absolute growth. And the problem of absolute growth isn't about different body proportions. It's about describing the geometry of the organism. So here you can see that you have a fish, and you have these different axes upon which you're measuring. You can measure this in different ways. Basically, you're trying to extract shape parameters from this object. Now, he uses a grid, but that's for a pre-computational era. In the computational era, we can actually describe shapes like this using a number of methods. So we can use, of course, polynomials, and the famous dictum by von Neumann that if you give me 5 parameters, I can draw you an elephant, and with the 6th parameter, I can have it wiggle its trunk, meaning that you can achieve any shape if you have enough parameters. And of course, in that case, you can fit a curve to the shape of an elephant, and you can have motion of the trunk and all of that in a relatively small number of parameters. So as long as we have these shape parameters, as well as they're well characterized, we don't need that much in terms of parameters or parameterization to achieve a description of the shape. And then, of course, it's absolute growth in terms of the organism. But if we were to compare this with another species, we would also have this similar set of shape parameters, and we could find sort of the things that distinguish those two species, which is useful information, especially if you're building a phylogeny based on phenotypic character traits. And so we can use a number, so we can use polynomials, we can use a Cartesian coordinate system like D'Arcy Thompson did, we can use other coordinate systems depending on the shape of the morphology, and then we can use conformal maps. Conformal maps are interesting because they allow you to warp the morphology and explore some of these shape parameters in more detail. You're basically building this homeomorphism between the absolute shape of the organism and some sort of conformal map that might describe an entire clade. And you just simply find the shape parameter values for your specific species on that conformal map. So this, there are different methods that we can use, methods that were, kind of developed well past the time of D'Arcy Thompson. Okay, so that's the problem of absolute growth. Now this is a graph where this is stating D'Arcy Thompson's vision of absolute growth, where we have the rate of growth versus acceleration of growth. And of course, this is sort of the first order versus the second order. So again, he's using growth as an absolute measure of sort of shape or geometry, and then he's treating this comparison as sort of like the equations of motion, where you have the first derivative of position, which is position, and then you have the second derivative of position, which could be velocity. And then of course you can have acceleration, you can have higher orders acceleration, and so forth. So this is rate versus acceleration, thought of as first order versus second order. And you can see it's very different from this graph, because this graph is described either by some linear function or more likely a logarithmic function where there's a saturation of head length versus body length. So you can't have a head length that's the equivalent of the total body length. That would be ridiculous. The head length is saturated somewhere where the rest of the body, because remember the rest of the body is also growing relative or changing relative to body length. And you have this sort of share of the body that the head takes up, and at some point it becomes too big to support by the rest of the body. Now in the case of absolute growth, however, we're not worried about that. We're worried about the rate of growth overall. So you measure this organism, you measure its physical parameters, and if they're growing or if they're positive, then that's where we're a rate of growth, and we can calculate that over time if we have a developmental system that's growing in size over time. So we can characterize a rate of growth, which is actually kind of a velocity of growth, if you will, because it's how fast, what is the change in growth over time. This is almost a time series, but it's just rate, so it's fine. And then acceleration of growth is sort of the second order of that, which means how does the rate of growth change? So you end up with a very different, you almost end up with a time series with a trace where you can look at these peaks and say that at this, at this rate, you have this acceleration versus the rate. And you can, know kind of, you can do, differential calculus on it. You can explore it using gradients, things like that. So we could actually do a lot of interesting mathematics with absolute growth that we can't do with relative growth, or at least directly with relative growth. So that's interesting. And people haven't really explored absolute growth as much as they maybe could. An interesting area. So this is hypermorphosis. This is where we have relative growth, but we also can characterize acceleration, which we're characterizing as the second order here. So instead of being peaks in a time series, this is a shift in the function describing this relative growth problem. So here we have body length and head length, as we do up in this graph. And we have our species here that form these trend lines, these linear trend lines. And there's a difference here, so that this trend line over time will increase. So it means that every time you look at a certain body length, and if you look at the difference between these two functions, the head length is longer. So the head length description for a given body length value is longer in this second function. And what does that mean? Well, that means that there's an acceleration of growth happening in the head, because as the body's expanding, the head's expanding faster. And so you can track this across this entire range of values. You look for any body length, we get a corresponding head length that's larger. And that means the head length is growing faster than body length, so a head length will take up a larger proportion of the body at, in adulthood when growth has stopped. And so hypermorphosis, though, this is just a different description of this acceleration growth that D'Arcy Thompson talked about in terms of absolute growth. And it's a little bit even more targeted because you have this relative growth. We know what the biological underpinnings of that are. They're not as vague as like absolute growth. I mean, if I described a shape of an organism, that doesn't mean anything necessarily. There are a lot of mechanisms that are interacting. But if I describe a relative growth problem, I can say this acceleration of growth is caused by X or Y. It could be a gene circuit. It could be a gene itself, just a single gene. It could be, all sorts of different, it could be something that you can target and maybe knock out or knock down in the genome and then look at how it affects the acceleration of growth. And, you can see that it might affect the acceleration of growth quite greatly. It might increase it, it might decrease it. So it gives you greater control. Nevertheless, this absolute growth is very interesting because it is sort of a way to look at growth is sort of almost like this fractal process where there's a growth in the entire organism. We can see the emergent features of like the genome and environment. But, so there are all these gene and gene environment interactions going on here that represent the shape. But then, you can also say, okay, there's an acceleration growth here at this point in development, or at this point in development, why? And it could be because of some phase in development. It could be because there are certain environmental factors. It could be that there are genes that are interacting at some point in development. So it's just a different way to characterize it. But I just wanted to point out there are important differences between the two. Okay, now there's this paper I found in Nature Methods. It kind of ties this together in terms of embryo models. So this is mapping morphogenesis and mechanics in embryo models. And this is from a group that is interested in physical forces, embryos, and they even get into organoids and things like that. So they're actually interested in cell differentiation in these multicellular structures that are differentiating and have some shape that we can describe, but also is due to geometry and forces. So you could say that they're kind of picking up the D'Arcy Thompson legacy, but also doing that, casting that in a modern way. So the abstract, recent methodological advances in measurements of geometry and forces in the early embryo and its models are enabling a deeper understanding of the complex interplay of genetics, mechanics, and geometry during development. They actually talk about flows and forces. So morphogenesis can be described very generally by tissue flow, that is the local rate of displacement. So this is due to growth or due to cell migration. Usually if you have more cells, you have more flow, you have more displacement, but not always. Tissue flow can be obtained from imaging data directly at the pixel level using particle image velocimetry or optical flow. In a first application of such techniques to embryo models, optical flow is used to demonstrate that the endodermal cap forms by collective cell migration in 3D gastroloids. I'm going to show you this in this figure 1. Alternatively, tissue flow can be obtained by cell tracking, which provides more information about cellular level movements that underlie the overall tissue change shape. So these changes in shape, these rates of change, are sort of affected by cellular level movements, migration, things we can track and figure out the time course of. They map to differentiation events in the embryo, and you can slow them down or speed them up based on cell doubling time and other types of things like how cells adhere to each other, how they form sheets, how they can sense their positional information, etc. Recent improvements of cell tracking include machine learning approaches that improve segmentation and efficiently handle very large data sets by automatically discriminating spatiotemporal features of cell lineages and cell division events. So this is where we have the spatiotemporal aspect of cell lineages and division events. We know kind of when cells are dividing, what lineage or sublineage they're in, and how that affects where they're located in the embryo. And then that gives us some idea of how shape, the sort of the precursors of shape. These methods were shown to be very effective at analyzing mouse gastrulation and can be anticipated to soon find application in studying embryo models. So this is, they're working a 3D gastroloid. And so they're showing some cartoons and they're showing some microscopy images. So we'll start at A. So A is a schematic of a 3D gastroloid. It has this bean shape and it has a red end here, which is the endodermal cap, which they mentioned before. And so gastroloid is not an embryo, but it's like this collection of cells that kind of takes on different shapes, primordial shapes mainly, but they're supposed to be differentiating cells or trying to behave collectively and do something functionally interesting. B is where we have tissue flow in a 3D gastroloid expressing brachyuria reporter, which is dark, and then the other cells which aren't. So you have these cells in the gastroloid that are expressing like these developmental markers and some that are not. So they're kind of being primed for differentiation or primed for a specific developmental stage. C is the fusion of mouse embryonic stem cell aggregates. The equation in C defines the expected time evolution and can be fitted to obtain the tau and beta parameters, which are proportional to the ratios of shear viscosity and shear modulus. So this is where we have this fusion of embryonic cells. We have this measurement of their time evolution in terms of how they're fusing together, and then you have these different parameters that relate to physical properties such as shear viscosity and shear modulus, respectively to surface tension. So this is not the same thing that Thompson was arguing, which is to measure the phenotype and look at how it gets warped by the grid. This is where we're using physical parameters and we're describing the shape changes in the whatever it is, this cell fusion in this case. And then we're describing the forces that act upon it. So we're not describing it just purely in terms of a coordinate system. We're using like shear modulus. And we can measure those with newer technologies. So this is why they're doing this. And it's actually a lot more informative and allows for a lot more prediction than just simply looking at differences in shape. D is the schematic of the ETX embryoid. So this is something they mentioned in the paper. This is another type of organoid. E is the tissue cartography, which is using IMSANE, which is image surface analysis environment. So instead of INSANE, it's IMSANE, and it's capital and small letters based on the acronym. So this is done for the inner epithelium, consisting of epiblast and extraembryonic ectoderm. So this is E, where you see the shape, the extracted 3D projection. In F, you have mechanical equilibrium, where interfacial tensions T and cellular pressures P, and an epithelium can be inferred from the vertex angles and membrane curvature radius. So you can see this unrolling process, where you can look at the shape of the cells and infer the forces. So it's not just about putting them on a grid where it's deformed. You actually can infer the forces from that because the forces operate by physical laws and they're predictable. And then, of course, this unrolled 2D projection using IMSANE, and it just basically, you have to use, of course, cell segmentation, very good cell segmentation algorithms, and then have this account of what the effect of different strains are on a cell's morphology. So if it's being squeezed or pulled apart or whatever. G is, actually F is mechanical equilibrium, interfacial tensions T and C. This is where we have this cartoon again, where we show at a trijunction this time, where these interfacial tensions and cellular pressures kind of play a role. So you look at this image and you see these deformations. And here we have a mechanical model where the deformations are translated into parameter values at the cellular level. And we know kind of what these are. We can measure these empirically. We can also use imaging methods like this to sort of understand how these things change, and we can model it this way. G is the inferred membrane tensions from cell geometry in Drosophila lateral ectoderm, so showing an example in Drosophila instead of an organoid. H is the schematic of a 2D gastroloid, so another cartoon. And then the map traction stresses in I of a 2D gastroloid. So we have the cartoon of a gastroloid here, and then this is the idealized one in H, and then this I is where we have the traction stresses in pascals, and we're measuring it. The colors are lighter is higher, darker is lower. And so another point they make is that you don't start off with the adult phenotype. So one thing that we didn't see in the examples from D'Arcy Thompson was that you have an embryo and you have a mature phenotype, and you can't really compare the two because there are a lot of intermediate stages. And with embryoids you can see this, because in embryoids you're growing them de novo. You're growing them from cells that are dissociated, and then they just grow in a culture, and so they form these aggregates independent of, like, say, an embryo, and then they form these shapes and these structures that have function. And so in early embryoids we can look at maybe some of the things that aren't constrained by embryos or aren't constrained by this sort of linear view of development as moving from like kind of a common embryo to differences. We have all these different dynamics that play a role in what it ends up looking like. We also have buffering, which maintains sort of a homeostasis in development. But basically, the embryoids, I think, are a window into what development could do. And so, anyways, so before forming large epithelial structures, embryo models typically combine small numbers of embryonic and extra embryonic cells in suspension. So this is where we're using embryoids to build this embryo model. We have these dissociated cells in suspension. We have small numbers of cells, and then they double in place, and they migrate in place, and they form these patterns. The initial dynamics of these cells as they rearrange into organized structures is of great interest. Understanding this benefits from analysis of individual cell shapes and interfaces in 3 dimensions with high accuracy in single cell measurements of mechanical properties. So again, we want to take this broad emergent view, but we can't do that just in one shot. We need to kind of go down a couple of levels and build models of individual cells interacting with other cells, and then understand those kind of interactions as contributing to these larger aggregates, and then eventually to the larger phenotype. Recent advances were made for subcellular segmentation of small embryos, including early mouse and Ascidian embryos, with the goal of more accurate inference of mechanics. Delaney watershed was designed to avoid introducing biases, so they used a Delaney watershed segmentation technique, which, depending on what you're trying to learn from the data, if you're looking at, say, a lot of angles, you want to, and a lot of the effects of forces, you want to have an algorithm that allows you to do that. Watershed apparently, or the Delaney algorithm, is good for that. A different approach generalizes the 2D approach for enforcing consistency of the segmentation with mechanical equilibrium to 3D. And then this is this 35. They just say it's a preprint. This is by Lemaire, so this is in Ascidians. This is the mechanical atlas for Ascidian gastrulation. So they've done this in, looked at mechanical stresses in Ascidians and built a mechanical stress map. And, this is all sort of in situ. This isn't like a organoid where they're doing this. Both methods then can be used to improve previous 3D force inference. To verify predictions, tension of and adhesion between individual cells can be measured directly with micropipette aspiration. This was previously done for the mouse blastocyst and recently applied to measure the differential adhesion between cell types in ETX embryoids. So this is just kind of thinking. And then, of course, we can talk about patterning and morphogenesis, where we haven't studied tissue patterning and morphogenesis in the same system, but now with these embryo models, these embryoids, we can do that. And so we can relate mechanics to tissue geometry, and that gives us sort of this full account of the Thomsonian view of these organisms in total.